So, we have finished the basics of protein and amino acids. Now, we come back to our normal class of that milk protein, right. We have finished milk fat and now we will go to milk protein. So, in our this dairy and food process and products technology class in lecture number 19, we come back to milk protein, right. So, in that milk protein when we are talking, so we have seen that proteins are made of carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus, right. So, that sulfur, phosphorus in some of the proteins it may or some of the proteins it may not be, but normally these proteins are characterized by the nitrogen content, and that is why we obtain the protein content with the help of the Geldal method, and there we get the nitrogen, and with the conversion factor we get the protein, right. And we have seen that proteins are made of amino acids with specific properties that are determined by the side chains of the amino acids in the polypeptidic chain. These all are now no longer Greek to us because we have completed, right. And conformation of the protein depends on the hydrogen bonds, hydrophobic interactions and salt bridges formed between peptide chains. All these salt bridge, hydrophobic or hydrogen bonds or peptide bonds, these also we have covered up, right. So, regular arrangements include beta sheets and alpha helix, right. So, this alpha helix and beta sheets also we have covered in our previous classes, right. So, that alpha helix where it is like a helical group, right. So, helix means it is as helix we know. So, this is like this and this is one and another is like that. So, this is like a helical group it appears, right, and all these depends on the temperature, ionic strength, pH that affects the protein conformation, and conformation we also said is nothing but the arrangement or the spatial arrangement in the spatial arrangement of the functional groups of the molecule, that is the conformation of a particular molecule or atom or system, right. So, this we have seen. Now, let us look into that. Normally, milk contains around say 2.8 to 4%, out of which 30 to 2.8 or to 4% is around say 30 to 35 grams per kg of the milk. It contains protein or this nitrogenous compounds and remains constant because normally proteins do not come out as fat we have seen. If we just keep fat just like that standing for some time, we have seen that fat molecules do come out and get separated from the milk, and because of that the fat component gets depleted, right. But unlike that, unless you are heating milk, this protein does not come out and protein remains more or less constant in the milk system, right. So, this is influenced by the lactation stage of the animal. So, this protein content also depends on the lactation stage, that a little we have, I may not be a little, elaborately also we have discussed earlier, right. So, two types of proteins are there, one is called casein, which contains around 80%, and lacto or lactalbumin, right, lactalbumin, or which is known as whey protein or serum protein. Now, what is serum, what is plasma, those things also we have covered earlier, right. So, this contains around 18 to 20% of the whey protein, of the total protein, right, and this some derived also proteins are also there. For example, lactoglobulin, which is maybe around 0.05 to 0.07% present there, right. Now, out of these proteins in milk, that is casein and whey protein, right, casein contains 80%, whey proteins roughly we call it to be 20%, 80% protein and 20% casein and 20% we call to be whey protein, right. Of what? Of the total protein present. To milk itself contains protein, we normally call it to be around 4%. Again, the many factors are responsible for this variation of the protein. So, we have discussed on that. So, assuming out of 4, 80% is casein. So, that means around 3 to 3.5 grams of protein or casein per 100 gram of this milk, and remaining is out of 4. So, remaining 0.5 to 1 gram is your whey protein, right. Now, this casein, there are 4 types of casein, they are having variation, 4 types. They are alpha S1 casein, alpha S2 casein, beta casein and kappa casein. So, I repeat 4 types of caseins: alpha S1 casein, alpha S2 casein, beta casein and kappa casein. These 4 types are there. These are phosphorylated and hydrophobic. These caseins are phosphorylated and hydrophobic, and they are associated with themselves and each other, right. So, they respond, they represent around 38%, 10%, 36% and 13% of the whole casein. That is, alpha S1 casein is 38%, alpha S2 casein around 10%, beta casein is 36% and kappa casein is roughly 10%, right. So, this is about the casein 4 types which are present. Overall, 21 variants of casein have been identified that occur by genetic mutation, right. Caseins have distinct disordered molecular structure that lack disulfide bridges. Also, caseins are very heat stable and withstand temperature about 150 degrees centigrade before getting dissociated, means it may get into your dissociation, means denaturing, right. So, denature also we said the other day. So, before it is getting dissociated or denatured or degraded, that it is around 150 degrees centigrade it can withstand, right. Hydrophobicity is due to this hydrophobicity is due to the high ratios of apolar amino acids. So, those amino acids which are not polar, hydrophobicity is due to that high ratios of polar amino acids. Apolarity is even in valine, leucine, isoleucine, phenylalanine and proline. So, they do have lot of apolarity in them, and that in turn is influenced to the protein, that is this hydrophobicity of the casein. Hydrophobicity of the casein is due to the apolarity of the casein molecule, that is the high ratios of apolar amino acids, in which we have seen that valine, leucine, isoleucine, phenylalanine and proline, they do call high ratios of hydroapolarity, right. And apolarity ranges between 35 to 45%, that is almost 50-50 is the polar and non-polar, right. And because of this non-polarity or apolarity, they do react, or they do behave like hydrophobic in nature, right. Now, this hydrophobicity can be encountered by high phosphate content, if there be high phosphate content, because phosphate is hydrophilic, right. Phosphate group is hydrophilic, very much hydrophilic. So, that will encounter the hydrophobicity if that is present. So, to nullify the hydrophobicity, lot of phosphate groups, if lot of phosphate group can be present in the protein, then the hydrophobicity of the protein can be reduced. That is one way to counter the hydrophobicity. Or low concentration of sulfur containing amino acids such as methionine and cysteine, that allows casein to be reasonably soluble in water, right. So, if low concentration of sulfur containing amino acids are also present, for example, they are present, we have seen in sulfur in methionine and cysteine. So, if small quantity of methionine and cysteines are also there, then also that can counter the hydrophobicity or hydrophobic nature of the protein, and it will not repel water to that extent. We have seen it is around 35 to 45% apolarity, that is hydrophobic or non-polar systems in protein, right. So, we come to then that the casein's susceptibility to proteolysis is due to the lack of secondary and tertiary structures of alpha casein and beta casein, alpha S1 casein and beta casein. Because of them, they do not have the secondary and tertiary structures so much. So, that is one of the reason why the proteolysis, that is breakdown of protein, proteolysis is breakdown of protein, that or casein, that casein is susceptible to proteolysis, that is breakdown of the casein is due to the fact that the alpha casein, alpha S1 casein rather, because we have not only alpha, but alpha S1 and alpha S2. So, out of that alpha S1 casein and beta casein, they do have less or they are lacking secondary and tertiary structure. So, that is why the the proteolysis of casein is possible, right. Now, what is alpha S1 casein or alpha casein, right? So, alpha caseins means alpha S1 and alpha S2 caseins. So, if you look at alpha S1 casein, that's alpha S1 casein is having 199 amino acids and is approximately around 23.6 kilodalton in length, right. Size, molecular size is 23.6 kilodalton. Now, like normally we say the lengths in millimeter, meter, then micrometer, then nanometer, but these molecules are being said in terms of Dalton, and they are around 23.6 kilodalton in alpha casein, around this is average, around 199 amino acids are present. Highest charge of all the casein molecules. So, it has maximum charge in all the casein molecules, and that is the reason why we said that when you have the freshly drawn milk, it may have the acidity, and some acidity may be imparted by this casein molecules, right. So, it has high charge, that is alpha S1 casein. This consists of least 8 this consists of least 8 phosphoserine units, right. List 8 phosphoserine units. It has 17 proline residues that ultimately disrupt the formation of secondary structures such as alpha helix, beta sheets. So, alpha helix, beta sheets are secondary structure. So, because of the 17 proline residues, they do not allow the formation of the secondary structures like alpha helix or beta sheets. This is the reason why alpha casein is not soluble or is just before this slide we had said in the previous slide that they are hydrophobic, right. The hydrophobicity is because of that. So, they are or they can be easily that is dissociated or degraded or denatured because of the non presence of the secondary and tertiary structures, right, alpha S1 casein. Whereas, alpha S2 casein, they are consisting of 207 amino acids and their average size is around 25.4 kilodalton, which was 23.6 kilodalton in alpha S1, here it is 25.4 kilodalton. So, least hydrophobic casein molecule. So, the hydrophobicity in casein, that is alpha S2 casein, is the minimum hydrophobicity, that is it is not that bad with the water system or polar system, right. So, it has least hydrophobicity amongst all the protein, amongst all the forms of the proteins, that is alpha S1, alpha S2, beta and kappa casein, right, and it has several genetic variants contain between 10 to 13 phosphoserine units. It contains between 10 to 13 phosphoserine units, whereas in alpha S1 it was around 17 phosphoserine units, right, and it contains 2 cysteine residues, right. It exists as a dimer in milk, right, not as a monomer or polymer. It exists in milk as a dimer, that is, two molecules are associated as dimer, right. So, looking at alpha S1 and alpha S2 casein, we then say that beta casein, that is other type of the casein, that contains around 209 amino acids and approximate size is around 24 kilodalton, right. It has the most hydrophobic casein molecule. It has the most hydrophobic casein molecule. The hydrophobicity, if you remember just some time back, we said is primarily due to alpha S1 and beta casein, right, and beta casein has the highest or most hydrophobicity amongst the caseins varieties, right. So, there are 6 known genetic variants that contain between 0 and 5 phosphoserine units, right. So, phosphoserine units are becoming less, much lower. Alpha casein was 17, alpha S1 was 17, alpha S2 was between 10 to 13, and this one less than around 5 or 6, around 5-6 numbers of not around 50 to 5 phosphoserine units are there. Similar to alpha S1 casein, beta casein has few secondary structures due to the presence of 35 proline residues, right. So, we said that the degradation or the proteolysis of the protein is primarily due to the lack of the presence of your secondary and tertiary structures in alpha S1 casein and beta casein. So, here also you see that it has a little, beta casein has a little few secondary structures which may help to sustain, which may help to resist a little against the proteolysis, right. So, the third one is gamma casein, okay, alpha S1, alpha S2, beta, that is alpha casein, beta casein, gamma casein, right. So, gamma casein is derived, if we do not take pen then it will go to the next page. So, alpha casein is derived by hydrolysis of beta casein by the enzyme plasmine, called plasmine. So, alpha casein is hydrolyzed to beta casein, gamma casein is derived by hydrolysis of beta casein by the enzyme called plasmine. So, from beta casein gamma casein is produced, and that is by the hydrolysis with the help of the enzyme called plasmine. And 3 variants have been identified near the carbon terminal and of the beta casein molecule, right. So, there could be 3 such variations of the gamma casein. Now, if we look at then we kappa casein, that is what is remaining, that kappa casein is what that kappa casein consists of 169 amino acids and approximately around 19 kilodalton. Approximately around 19 kilodalton is the size, and it contains both glycosyl and phosphoryl, that is, it is glycosylated and phosphorylated residues it contains, right. So, glycosyl and phosphoryl, these two groups. So, that means it is glycosylated as well as phosphorylated. So, it contains both the glycosylated and phosphorylated residues, right. Now, this is the unique of the kappa casein. It consists of it can exist as a dimer up to a decamer with the subunits held together by disulfide linkage, right. Disulfide linkage, the other day we had said that this is like this, right. So, we gave it to be with chlorine and many others. This is the simplest S2Cl2, right, simplest disulfide which we can think of or which we can give an example. So, that disulfide linkage is subunits held together by the disulfide linkage, right. Then, unlike the other caseins, it is not sensitive to calcium and surrounds the micelles, keeping them intact. So, micelles we have already said earlier. Now, again it may be in the subsequent class we can, since now we are again in the milk protein. So, in the casein, so casein micelle maybe a little we can highlight, but earlier we have said. So, unlike the other caseins, it is not sensitive to calcium and surrounds the micelle, keeping them intact. It usually contains one phosphorin unit. However, genetic variants containing 2 or 3 phosphoserine units have been identified. Further, 9 variants have been identified that demonstrate different degrees of glycosylation, right. So, we have come across with different causes why proteins are hydrophobic in nature, which component of the protein alpha, beta, gamma and kappa, out of which alpha has alpha S1 and alpha S2, how they are behaving, what is the reason, how many number of amino acids are present, all these we have discussed, right. So, next we may be going to the casein micelle. Okay, thank you.